Good afternoon, everybody. My name is Alvaro Mata. I am professor of biomaterials and biomedical engineering at the University of Nottingham, and I am a member of the board of the Biomedical Engineering Division at the Institution of Mechanical Engineers. And it is my great pleasure to host this year's Duncan Dawson's Memorial Lecture from IMechE. This year, we have the great pleasure to have Professor Rebecca Shipley as the speaker. Rebecca is professor of healthcare engineering at UCL and director of the UCL Institute of Healthcare Engineering. I will let her tell us about her work, but wanted to take the opportunity to share a few of her impressive and impactful achievements. So let me just start with the last couple of years. In 2016, she received the Rose Trees Trust Interdisciplinary Prize for research at the interface of mathematics, computer science, and medicine. In 2017, she co-founded the UCL Center for Nerve Engineering, which brings together physical engineering, life, and clinical scientists to tackle nerve injury repair. In 2020, Rebecca co-led the UCL-Ventura program to manufacture non-invasive breathing aids at the scale for treating COVID-19 patients. The team secured regulatory approval within 2 weeks, leading to the manufacturing of more than 10,000 devices for use in the UK, and now devices are actually in use in at least 15 countries. The team was awarded the Royal Academy of Engineering President's Special Award for Pandemic Service in 2020 and the Health Service Journal Awards 2020 Acute Sector Innovation of the Year. For this, also, Rebecca was awarded an OBE in the Queen's Birthday Honors. Now, before we call Rebecca, I do want to let you know that you have the opportunity to ask questions in the Ask the Question box. And I will be reading the questions at the end, but feel free to write the questions as the presentation moves along. This is a 1-hour event, I want to remind you. So with that, we are ready for Rebecca. Rebecca, it is my pleasure to have you. Thank you for joining us today.
Thank you so much for the very generous introduction and also for the invitation to speak to you all today. It's a real privilege. Thank you very much. Hopefully my slides are working okay. So so Alvaro's given an introduction to my roles. I'm based in the Mechanical Engineering Department at University College London, and I also run our Institute of Healthcare Engineering here. And I'm going to start my talk with a very broad perspective and then talk some specific exemplar activities that we've been running around healthcare technologies. So globally, there's a real need for new healthcare technologies, and this is a topic that I think the COVID pandemic has really shone a light on. Of course, the needs were in many cases already there, but I think it's really raised our awareness of some of the global challenges relating to health and the role that we can play in working together globally to try and tackle them. The United Nations Third Sustainable Development Goal is ensuring healthy lives and promoting well-being at all ages, and that really sets out, I think, the global challenge. And of course, the pandemic has interrupted much progress that's being made. There's a real diverse set of exciting areas and problems to work in, and they can span things like prevention, monitoring, for example, thinking about mobile phone apps and monitoring public health, through to technologies for diagnosing disease, treating them, and then also rehabilitating patients who have experienced trauma and disease. Of course, some technologies are very commonplace in our lives now, things like wheelchairs or x-rays or ultrasound machines, step counters even, that so many people now wear on their wrists on a day-to-day basis. And then there's the more kind of cutting-edge disciplines that are really advancing and growing, things like nanoimaging, AI, surgical robots. There's an extensive list that we could talk through. But in my mind, one way to kind of break them up is to think about more fundamental research and more translational research. And I think we really need a balance of the two. Fundamental research can often help us to better understand both physiology and pathology or disease. And I think to do that really requires teams of people working from a range of different disciplines together, like biologists, chemists, physicists, engineers, mathematicians. And then the more translational part is around developing technologies that are fit for purpose and can be deployed into healthcare systems. And I think that really requires end user engagement, whether that's working together with doctors, nurses, healthcare professionals, but also patients and the public and their families. And then thinking about things like regulation, enterprise mechanisms, and of course the list goes on. So I think common to both those areas, as I mentioned, is this need for working collaboratively with people from a range of different expertise in a multidisciplinary fashion. And I think that aspect really underpins the various different projects that I run and am involved in. So today I'm going to talk through a couple of examples that are really kind of sit at the two ends of that spectrum of fundamental research through to deployment, to hopefully give a flavour of some exciting projects that I think are really relevant in the area of biomechanical engineering, but also hopefully a perspective on some of the kind of perspectives on running multidisciplinary projects, and I think the importance of that diversity of expertise and input into problem solving. So the first case study I'm going to talk through today is around quantitative modelling in cancer diagnostics and therapy. And this is an area that I really started working in when I was doing my PhD. So I trained as a mathematician, an applied mathematician at the University of Oxford, and my PhD research was in developing multi-scale mathematical tools for linking tissue properties on different length scales, and I did a lot of that work around cancer, and it's an area that I've continued to develop and grow throughout my career so far. Since I've been at UCL, I've had a really strong collaboration with Simon Walker-Samuel, who's a professor here in biophysics and imaging, and he's based at our UCL Centre for Advanced Biomedical Imaging. And with each of these case studies, I'm going to emphasise the team composition up front, because I think that's one of the aspects that I think I find really interesting and important, and as I said, really kind of shapes a lot of the work that I do. So Simon and I have been working together for, I'm not quite sure, I dread to think how long now, probably not sure till the decade. And we've sought to build a team together that really brings together the different expertise that you need to tackle some of the really relevant and important questions in understanding cancer growth and development and how we can diagnose and treat it. So there's some examples of the people here that have contributed to the work, but some of those people are mathematicians, some of them are physicists and biophysicists, some are chemists, and then we work with biologists and a team of clinicians as well. And I think that diversity of expertise is really important in meaning that the pertinent questions that are relevant from a diagnostic and therapeutic perspective are well understood and inform the work that we do. But the work we do is often quite fundamental. So it's developing new imaging techniques, it's integrating those with mathematical and computational models, and new approaches, for example, from chemistry and biology. So I'm going to start by just talking a bit about the role of the circulation in your body. So I'm sure everyone is familiar with your circulation, but essentially we have a blood vessel network that runs throughout your body, and its role is to, in a simplistic sense, bring oxygenated blood within a very small distance of every cell within your body so that you can meet metabolic demands. And obviously that oxygen is supplied through your lungs, and your heart pumps that blood around the body. Blood vessel networks have a really interesting structure. So we have we have large vessels that are feeding vessels like arteries. They branch down into smaller vessels called arterioles, and they in turn connect into a capillary bed in each tissue, which has a topological shift. So it's no longer branching; it's more of an interconnected mesh structure, and that's the level of the vasculature at which gas exchange occurs. So oxygen, for example, will diffuse out of the vessels into the surrounding tissues to feed the cells of your body. And then there's a mirroring drainage system that's comprised of venules and veins. But of course, different tissues in your body have different metabolic requirements. So, for example, your heart has quite a high metabolic requirement, and if you exercise, that will obviously increase. Whereas other tissues, to pick one example, cartilage has quite low metabolic requirements. So as a consequence of that, vascular structures in your body are tissue specific, but they can also change in time. So that could be through changes in metabolic demands. It could also be in response to injury or disease or repair. So I'm really interested in understanding what these vascular structures mean in terms of how blood and solutes like oxygen, drugs, therapies are delivered to tissues in your body, and understanding how we can link that in with both diagnostic tools like imaging and into optimizing how we deliver therapies for various different diseases. And obviously blood flow modelling can provide a really useful tool in that process, both in terms of understanding and predicting what healthy and pathological or diseased tissues look like, but then also in integrating with data from other sources like imaging to provide better diagnostic or therapeutic strategies. So this really gives a bit of a summary of blood vessel structures. And I just kind of mentioned this, so I won't go into too much detail, but on the right-hand side here, you can see different scanning electron micrographs from different tissues in your body, and it gives you a feel for how the structures really are highly tissue dependent. And not only are they tissue dependent, but they can also vary quite significantly within an individual tissue. So this slide here really summarizes some of the work that we're trying to do here. So if a cancer patient, for example, presents at hospital and is being investigated for cancer, so at the diagnostic end of the spectrum, there's a range of different ways that that might be tackled. But the kind of classic scenarios are that that person might, depending on the tumor type, be biopsied, which is where you remove a small sample of the potentially diseased tissue to look at under a microscope, and that gives you really high resolution information, as you can see on the left hand side here, about things like cells, cell structures, cell shapes, and the kind of structure of the surrounding tissue. And then that individual might also have imaging, so magnetic resonance imaging is one example, but there are others obviously that are used in hospitals in commonplace. That's non-invasive, enables you to do that multiple times, but gives information on a much coarser length scale. And one of the challenges that we're interested in trying to address is what's the link between those data? So how can you link the really fine scale information that you might get from a biopsy to the kind of data that you might get non-invasively using imaging, and use both to better understand the pathology of a particular tissue and how you can then take that forward for treatment. And this really gives an example of one of the challenges of treating cancers that, again, mathematical modelling and imaging can play a role in trying to contribute to, which is that cancers often have particularly chaotic structures, vascular structures, and that's compounded with a really often quite abnormal microenvironment and lots of other properties of the tissue, like a lack of functioning lymphatics, which compound to mean that transport of blood and therapeutics that might be delivered in the blood are highly challenging. So this is an example of a cross-section of tissue where a chemotherapy drug called doxorubicin has been delivered through the blood vessels. You can see the blood vessels are coloured in red. The green stain represents hypoxia, which is essentially oxygen deprivation. So essentially they're regions that are residing in a low oxygen status, and that's essentially a balance of the diffusion distance for oxygen versus how quickly it's metabolized by cells. And then the blue regions are essentially where the drug has gotten to. And as you can see, the drugs will often collect in the nearby regions surviving vessels, which are the regions that they get diffused into. But then because often tumours have quite large intervessel separations, it will mean that there's often quite wide regions of the tissue that the drug can't access. So again, there's the opportunity to combine approaches from, for example, imaging and modelling to better understand these processes and then optimise the properties of drugs and how you deliver them to better treat cancers. So this really zones down now on the specific example that I'm going to talk through in the next 10-15 minutes, which is around the kind of data that we can now get access to understand the structure and function of tissues. And this is where we're kind of moving into more fundamental research questions to some extent. So hopefully I've made the case that it's really useful and important to be able to understand the structure of tissues and use that to inform the kind of tissue scale functional information that you might get, and use both diagnostically and therapeutically for cancer patients, for example. And that has obviously motivated, among other motivations, different multi-scale imaging methods to look at what tissue structures are at really fine scales. And as I said before, I'm going to talk through really some of the work that we've done to understand the linkage between data on these different scales and what we can do with them. So Simon Walker-Samuel's group and our joint groups have been developing different optical methods to look at these fine scale structures. And I'm going to give an example of a specific one, which is around 3D optical imaging, which the objective here is to allow us to essentially digitally reconstruct the blood vessel structures of whole tumours to use as the substrate for modelling to understand how blood and therapies get delivered to these kind of tissues. And the challenges here are being able to resolve information at really fine scales. So the smallest blood vessels in tumours and most tissues, in fact, might be of the order of a few microns in diameter. And if you want to think about what to do for a whole tissue, like a whole tumour, then you need that kind of information for that whole structure. And, a whole tumour can be millimetres, centimetres in size. So there's a real challenge in terms of the imaging technologies and then the image analysis and computational technologies that go alongside analysing these kind of data sets. So this is an example of a colorectal tumour from a cell line. So it's LS174T cell line, which has been grown up in an animal model. And here, as I said, 3D optical imaging has been used to look at the structure of the blood vessels. Essentially, the blood vessels are all labelled with fluorescent lectins. The tissue is then optically cleared, and then it's imaged using optical projection tomography, or OPT. And then we do quite a lot of processing on those imaging data to enable us to reconstruct the tissues. Essentially, that's done in very, very fine slices, and the structures are reconstructed in 3D computationally. But what's really nice about this is that it has enabled us to recapitulate vascular structures for whole tissue samples. As you can see here on the right, this is one of these cancers, so it's one example. You can see around a millimetre length scale, so the whole tumour is centimetres in size, and then the smallest vessels that we've picked up are down to a few microns. This video, which is hopefully playing okay, gives you hopefully a feel for the kind of scale of the networks that we can now get access to. And I mean, these are beautiful networks. They're intricate. You can see the heterogeneity that I talked about as you move through the tissue sample. Properties like diameters, vessel lengths, densities, intervessel separations vary quite wildly across the tumour sample. So there's an opportunity here to use these kind of data to inform the tissue scale imaging that's done as standard. So to address this, we've put together this framework that we've called REANIMATE, or Realistic Numerical Image-Based Modelling of Biological Tissue Substrates. And this is really geared at integrating at every step the imaging and the computational modelling approaches that we can use to tackle the kind of questions that we're interested in. And I think what I really want to emphasise here is that to do this kind of thing, it's really important that the imaging development and the computational modelling development are done hand in hand and aren't developed separately and then brought together at the end, if that makes sense. Because obviously it's highly important that the models are calibrated and validated, and that requires specific steps in the imaging sequences to enable that to be done. So essentially what we do is that we grow up a tumour in an animal model, so that's an in vivo model. Before that tumour is excised for optical imaging, that's an ex vivo imaging technique. What we do just before that is in vivo multimodal imaging, which is where you do, for example, the MRI imaging to capture the kind of data that you would get clinically from an MRI, for example, if it was done on a human. The animal is then sacrificed, the tumour is excised, and the high resolution optical imaging is performed to get the high resolution vascular architectures. So we image all of those features in 3D. We do a lot of work on those structures to segment and analyze them so that we can then reconstruct the full 3D vascular networks computationally. We then simulate the flow of blood and drugs, for example, through those vascular networks. So those geometries are used directly as the substrate for the models so that we can predict how blood flows through the tissues and how, for example, drugs are delivered. And at that stage, there's a really important validation and parameterization step where we compare the results of the modeling against the in vivo imaging. And that's a really, really, really pivotal step that's really challenging, but is very important to make sure that the predictions from this combined approach are predictive and quantitative. And at that point, if we have confidence in the approach, we can then use it to simulate delivery of drugs and optimize how that might be done. So this summarizes the kind of frameworks that we've put together, and I'm not going to talk through them in detail, but it's just to give you a flavor of the different kind of packages. So there's the biomedical imaging, which is separated into ex vivo, so outside of the animal, and in vivo imaging. There's various different components to the computational models incorporating prediction of flow through these really complex vascular networks and the surrounding tissue. And then there's delivery of solutes like drugs, and they're obviously all coupled, and the integration between the computational models and the imaging is really pivotal, both for calibration of the models and validation. These are the kind of simulations that you get out. So these are different simulations of the pressure distributions in these vascular networks, the blood flow distributions in the vascular networks, and then the interstitial velocities and pressures, which are the flows outside of the blood vessels in the surrounding tissue, so in between the cells and matrix, etc., that make up the rest of the tumor. So you can see that we can develop simulations that, you know, look nice, but are they accurate is the big question, and how do we make sure they're accurate? So to do that, obviously, we as I've already mentioned, we've made sure that the imaging and the modelling is well integrated. So we've included dedicated steps to essentially compare the imaging and the modelling predictions. So here you can see on the left-hand side of each of these panels, the perfusion in vivo imaging that was done in the animal. So that's the imaging data that was acquired. And then you can compare that against the counterpart predictions of the REANIMATE framework of the computational models. And we've done this for a range of different tumour types and repeats of each tumour type. So these two examples are both for colorectal. They're different colorectal cancer cell lines, so LS174Ts and SW1222s. And these two different cell lines, they're both colorectal, but they have very different vascular signatures, if you like. They're very different in terms of heterogeneity and vascular density. And at this stage, you can only really see the qualitative comparison, but you can see that it's pretty good. And then once you've got agreement, the next stage is that, as I said, you can use these kind of platforms as a tool to explore how drugs will be delivered to tissues. So this is an example of a bolus injection. So it's an injection of gadolinium DTPA, which is a contrast agent that's used as commonplace in imaging. And as you can see, we can now predict how that moves through the tissue, what kind of time course it follows, which regions of the tissue get preferentially supplied, and we can link that back to properties of the vascular architectures. Again, at this stage, once we've done this kind of solute delivery simulation work, we can then compare the in silico, so the computational results, to the in vivo results. And again, here are the examples. So this is essentially on the top, the simulation results, on the bottom, the measured in vivo results, and this is for a slice. These are for slices through the tumours, and essentially we can compare them directly. So broadly speaking here, you can see that a lot of the spatial heterogeneity that's imaged is captured in the computational models, and that the concentration distributions look about right. And then you can also pull out, obviously, the exact concentrations at different cross-sections through the tissue and compare them directly. I'm not going to talk through it in detail. If you're interested, you can go and have a look at some of the papers. But, you know, we're pretty good at recapitulating the distributions as you move through the tissues and over time, which, you know, is highly challenging because of the range of different processes and the real world context of the data that we're working with. So with some confidence now that this kind of platform is appropriate and quantitative in predicting therapy distributions, we can now look at what that means for specific therapies. So this is just one example that, again, I'm not going to talk through in detail, but all the details are in our papers, that's looking at vascular disrupting agents. So these are agents that are delivered to cancers to essentially change the properties of the vascular networks to make the tumour more amenable to other forms of treatment, so either delivering chemotherapy or radiotherapy, for example. And we've been developing the technique further to be able to look at how the vascular structures vary in time and to be able to correlate data on vascular architecture to basically which vessels get targeted by these kind of therapies, and then to optimize the dosage and the timing of subsequent treatments. So that is still work in progress, and we're expanding this to look at other therapeutic scenarios beyond kind of chemotherapy drugs, also nanoparticle mediated delivery of drugs and physical therapies too. So that's the kind of close of that first example. Hopefully it gives you a flavour for some of the work that we're doing in this space, and hopefully as well the need for these kind of multidisciplinary teams and work that's genuinely integrated from the outset to make progress. Right, so I'm not going to talk through this example in detail, but I wanted to just mention it because it's a similar kind of platform of work that we've been developing at UCL, which in this instance isn't focused on cancer, but rather nerve injury. So this is similarly work with a collaborator, so Professor James Phillips, who's a professor of regenerative medicine, and he's based at the UCL School of Pharmacy. And although the application area is very different, there's a lot of commonality in the underpinning disciplines and technologies that are needed to make progress in terms of treating people with nerve injuries. And so we set up the UCL Centre for Nerve Engineering to really bring together this multidisciplinary community at UCL and beyond, so spanning biomaterials through to computational modelling, imaging, through to cell and gene therapies, pharmaceuticals, tissue engineering. But what's really important here as well is that we work very closely with the end users that I was talking about at the beginning. So, for example, we run patient engagement events where we work directly with patients who have experienced nerve injury to understand their experiences and their priorities in terms of treatment, and also hospital groups. So the Royal National Orthopaedic Hospital and the University College London Hospital, we work directly with clinicians who are kind of embedded in the centre and help to define the research directions that we take. So if anyone is interested in finding out more or indeed collaborating in this area, then I would be delighted to hear from you. The second case study that I'm going to talk through now is at the kind of opposite end of the spectrum from my introduction at the beginning, in terms of it's a project that we've run in the last year in response to the pandemic that's very focused on delivering technologies into the healthcare system as quickly as possible. So I thought that these two examples would be quite illustrative, hopefully, in terms of the kind of range, I think, of approaches that we need across that fundamental to translational spectrum. So this is the story of our UCL-Ventura CPAP project. A CPAP is a non-invasive ventilator, And essentially, this is a program that we brought together extremely quickly to respond to the pandemic last March. So instead of talking through lots of the technical detail of this, I'm really just going to talk through the story of the team, how we came together and what we did, and where we've got to. So if you will be generous with me and allow me to take you back to March 2020. So obviously the pandemic had begun. Cases had been ripping through China and then Italy, and then we'd had our first cases in the UK, and it was very clear that we were going to follow a similar trajectory to these other countries across the world. So if we go back to mid-March 2020, the UK was looking at a crisis situation. There were many different models at the time that were looking at how COVID would spread through the UK population and what would be the consequent risks and requirements on the NHS. And at the time, there was a model need for up to 40,000 ventilators. Now, ventilators, mechanical ventilators, are devices that are used to treat the very most sick of COVID patients, or indeed patients with other respiratory problems. So they're managed in intensive care. But at the time, we had only 3,500 intensive care beds. And in the image on the right hand side here, you can see where we sat kind of internationally in terms of our number of critical care beds per 100,000 inhabitants in the UK compared to other countries, and we are relatively low on that list. But of course, there was also a worldwide shortage of ventilators and indeed most medical kit that you need to treat COVID patients, because obviously every country was ultimately facing similar challenges. So we potentially had a very significant problem. On the 16th of March, you may remember, but the UK government set out the ventilator challenge, and that was essentially aimed at corralling the UK engineering and manufacturing community into mass manufacturing ventilators at real pace for the NHS. But I think it was clear that this was going to be massively challenging. First of all, it was potentially unrealistic to expect non-medical companies, so that weren't certified as medical device manufacturers, to make sophisticated ventilators within weeks. The peak of the first surge was due to hit London by the Easter weekend, so it was genuinely weeks. Plus, an ICU bed isn't just a ventilator. One of the biggest limitations that we have is the number of trained ICU staff that are available to actually support people on these devices. And normally a person on a ventilator would have one-to-one care from a highly trained intensive care nurse, and obviously you can't upscale that kind of skill within weeks. So we started thinking, were there other options and ways to tackle this problem? So an answer would be to try and see what we could do to keep patients off ventilators. And so basically, I already had a collaboration with the intensive care unit at University College Hospital, which is one of the biggest teaching hospitals in London, and in particular with Professor Mervyn Singer and Dave Brearley, who are both critical care consultants there. I've got an EPSSC project, which is on modelling vital signs data in ICU, so pretty different, but we already had this collaboration set up. Now, Mervyn had been talking to colleagues in China and Italy to understand what their experience had been of treating COVID patients. And they found that when they were mechanically ventilating as many patients as needed advanced respiratory support, then their hospitals very quickly became completely overwhelmed because, as I said, there's a very high burden on support for patients on these devices, and also people can be on ventilators for weeks and weeks for COVID. So it very quickly overwhelmed hospitals. And the experience in China and Italy was that if they were treating patients with CPAP instead, then that could alleviate around 50 to 60% of patients from progressing to needing mechanical ventilation, which was good both for patient outcomes, but also for optimizing healthcare resource. So our position was, let's try and keep as many patients off ventilators using CPAP, but of course, We didn't have many machines in the UK, and there was an international shortage, international supply chains were breaking down. So could we make our own very quickly? So what we decided to do was to go back to an old device that was no longer being made. This is the Philips Respironics WhisperFlow CPAP device. It's out of patent, and but had previously been CE marked. Now it's a purely mechanical device, so it's quite simple to operate, and hopefully would also be simple to manufacture. And because it had been previously CE marked, there was the hope for rapid regulatory approval, which is obviously a pivotal step in ensuring that any medical device is safe and effective for use. So then the question was, could we make it quickly and at scale and during a global lockdown? So to tackle this, we formed very quickly a team that brought together the range of expertise that I think we really needed to tackle this kind of problem. So we needed engineers and mechanical engineers. So some of you might know my colleague, Professor Tim Baker, who's a professor of mechanical engineering design, and we're both in the UCL Mechanical Engineering department together. We needed healthcare expertise. So we needed the critical care teams who understand how these devices are used on wards in hospitals, and we needed manufacturing capability. So we put together basically a team that brought together UCL engineers, University College London hospital critical care expertise, and Mercedes high-performance powertrains, who are the arm of Mercedes who design and manufacture the Formula One engines for the Formula One team. And basically we brought this team together overnight and got stuck in. So I'm just going to talk you through essentially the time frame of what we did and where we've got to, and then I'll open up for questions. So the need for CPAP was defined in early March. As I said, we had weeks if we were going to be able to contribute. So we brought together this team and started working. So the Mercedes teams joined UCL. They turned up at UCL on the 18th of March and we bunkered down. We got an old device, one of these devices from the UCLH Anesthesia Museum, and we bought another one on eBay and basically reverse engineered it. So took it apart, redid all the internal drawings, did all of the material characterization, and long story short, we're able to produce the first prototype was made by Mercedes on the 22nd of March, so within 100 hours of the first meeting, and they turned up at UCLH for us to start testing in the hospital. That was plugging it into all of the hospitals on the different wards and testing them out on healthy volunteers. At the end of March, so 24th of March, the NHS issued guidance that included CPAP on the care pathway, and we got a first contract from the government to produce a small number of these devices. So the first 100 hours involved a lot of kind of core mechanical engineering work, really. So engineering design, CAD design, simulation work, CFD work, and was really exciting. It also really emphasised the need for this collaboration with the hospital. So we were going between the engineering units, if you like, and then we're straight, the hospital's just around the corner. So we were then in the hospital, in the wards, plugging these devices into the oxygen ports and testing them out. And I think that ecosystem that was already set up essentially and that we could just tap into was really pivotal in allowing us to make fast progress. So obviously regulatory approval is pivotal. So we submitted to the regulator. We'd been in communication with them from the second day, and we received regulatory approval for our Mark 1 device on the 27th of March. That was 10 days after we started. We then began clinical evaluations at both UCLH and also sister hospitals across London. And then ultimately, the Department of Health commissioned us to manufacture 10,000 devices as quickly as possible. But the next big challenge that we faced was essentially national hypoxia. So given the volume of patients that were turning up at hospitals, there was a very real threat that hospitals could run out of oxygen. And in fact, in the wave 2 over January and February, there were multiple reports of hospitals that got extremely close. So CPAP devices are typically very oxygen hungry. So there's a real issue here that if you treated too many people on a CPAP device, then you could compromise the oxygen availability for other patients on other kind of forms of respiratory support. And in fact, you wouldn't be able to treat as many patients as you might be able to. So in response to this, we did a lot of work on a Mark 2 device to minimize its oxygen utilization. and we brought it right down, essentially. So we did a lot of work, both on the breathing circuits, which are the tubing, the masks, the valves that connect the device to the patient, and also on things like the design of the air entrainment port to minimize oxygen utilization. And we were able to bring it down by about 70%, which made a big difference. So this Mark II device is the one on the right-hand side here. On the left-hand side, you can see the original and the Mark I device, which was the exact mimic. We submitted that to the regulator and got that approved a few days after the Mark I device, and that's the one that we took forward for mass manufacture. So ultimately, we then partnered with a logistics company to distribute these devices throughout the NHS and delivered the full 10,000 devices on the 15th of April. Mercedes HBP were phenomenal. They were the manufacturer. At the maximum rate, they were making about 1,200 a day. Ultimately, over the next few months, we ended up with 6 different MHRA approvals, not only for the original CPAP devices, which are on the top there, but all of the other bits of kit that are needed to go with it, because there was a supply chain issue globally at the time, so you couldn't get access to things like oxygen monitors, hoods, masks, that kind of stuff. So we ended up partnering with other actually small companies like Oxford Optronics and Life Racing, which are either medical or motorsports based, to again design and mass manufacture other pieces of kit. So where did we get to? So the devices have been delivered to 130 NHS hospitals across the UK and also overseas territories like Jersey and Montserrat to augment existing supplies. My apologies, this video isn't working, but it's a video of a patient that was featured on BBC News who was on one of these devices. So obviously we were delighted to be able to contribute in some way to supporting hospitals in their response to the pandemic. And this is very much ongoing work. Obviously, we're thankfully in a situation at the moment where hospitals are more under control, but I think it's there's a risk that we end up in difficult situations again, and these devices remain available and in fact are still in use at the moment. Did it have an impact on outcomes? These are data from the Intensive Care National Audit and Research Centre, which is the national data on intensive care. And there's quite a big shift in outcomes, particularly over wave 1. So here we're looking at data on the left, which is patients admitted into intensive care after the 1st of September last year, and on the right it's up to the 31st of August last year. And essentially over that wave 1, there was a big shift in how patients who were equally sick were treated. So the percentage of patients that were mechanically ventilated went down from 72% to around 50%, so that's a really massive reduction. This is for equally sick patients, and which basically that meant that number of patients were being treated using CPAP instead of mechanical ventilation. The duration of time, so the length of stay on critical care, halved, and mortality rates significantly dropped as well. So the last piece I'm going to mention in the last couple of minutes is around our international work. So it became very clear very quickly that, of course, a pandemic is global by its very definition. So there was a need in many other countries for a simple device that was purely mechanical, didn't rely on an electrical power supply, that could be used in a range of different healthcare settings, and was quite simple to operate and train healthcare workers to use. So in response to this, we released all of our designs open source. So essentially through a zero-cost license, alongside all of the manufacturing instructions, so that they could be downloaded to set up local manufacturing hubs in other countries. Now, this has been incredibly, we've learned a massive amount. I've learned a massive amount about global health and global healthcare. The designs have been downloaded over 9,900 times across 105 countries. We put together a whole support package that spanned the technical components, but as well as the clinical components and regulatory support. And we also established a UK hub to supply all of the subcomponents of the devices, because supply chain issues remain a massive issue for many countries, in particular lower and middle income countries. Of those 1,900 downloads, there's about 20 teams, so a very small proportion of those, the total, have made it through across the whole spectrum of download, design, testing, regulatory approval, mass manufacture, and deployment into hospitals. And some of these teams are still very much progressing. But the ones that have made a lot of success, we've worked very closely with. There are just the most formidable, amazing engineers and clinical teams across the world, but including in countries like Mexico, Peru, Paraguay, India, Pakistan, Philippines, and South Africa. And then the other piece that we've done is to work to supply devices directly to countries that don't have that local manufacturing capability. Obviously, many countries can't respond in terms of local manufacture fast enough to meet the urgent need of the pandemic. So we've worked with charities such as the International Medical Education Trust, who have sent devices to 5 hospitals in Palestine and 2 hospitals in Uganda, one of which was to set up their first intensive care unit. Mercedes-Benz South Africa donated 1,000 devices, which have gone to 90 hospitals across the East Cape. And then actually more recently, the UK government donated 1,160 of these devices to India and 130 to Nepal. So there's been a lot of activity globally, but there's also been I've learned a lot about the challenges associated with medical device development, manufacture, and regulation globally, which is probably a whole lecture in itself. But if anyone is interested in those areas, again, I'd be really delighted to talk to you about it. These are some photos of some of the teams, which I think really brings it to life. So this is a patient on one of these devices in India. Teams in Paraguay are still extremely active and are starting mass manufacture any day now. We've had a lot of contact and work with teams in Latin America, so Mexico here as well, and also teams in Ecuador. And then groups in Pakistan and India have actually set up mass manufacture and are supplying neighbouring countries, which is fantastic. And then in Uganda and Palestine, these are devices that were donated working with the International Medical Education Trust. If you're interested in this work, then these are a couple of papers that you could read about the story. And the last thing I just wanted to mention is that this work kind of all happened through our UCL Institute of Healthcare Engineering, which is an institute that I run at UCL and is dedicated to really interfacing our engineering community with our medical and clinical sciences communities, both across UCL and partner hospitals. So we work in lots of different areas. I mentioned themes earlier: prevention, monitoring, diagnostics, therapies and interventions, and rehabilitation and assistive technologies. And we're developing these kind of approaches in different areas. So a big priority area for us at the moment is around healthy aging. And we've got an Age Innovation Hub, which is live at the moment and is essentially aimed at engaging people from outside the university and defining the kind of technologies that are needed to support healthy aging, and then in developing those technologies further. And that's very much open to collaboration from people from outside of UCL, so other academic institutions, but also industry and people, anyone essentially that has lived experience of the challenges of healthy aging. So if you're interested, please take a look and we'd be delighted to hear from you. Thank you very much. It's been a real pleasure to tell you about some of the work I've been doing, and I'd be delighted to take any questions and be in contact with anyone after the event as well.
Thank you very much, Rebecca. It's fascinating and inspiring. It's wonderful to see the bridging that you do. You know, you're bridging disciplines, you're bridging modeling with experiments, bridging fundamental science and practical technologies and applications. And it's clear to see the impact that bridging has. So wonderful work, and thank you for sharing it with us. I have a few questions. Let me start from Huidong, who asks, from the tumor imaging of the geometric data of blood vessels, are there any chemical, biological, transportation, or reaction phenomenon incorporated in the computational fluid dynamic simulation?
Yes, there are. It's a really good question. Thank you for it. Yes. So essentially, we do the blood flow modelling. So we model the flow of blood in the blood vessels and in the surrounding tissue, and then we layer onto that solute transport model. So that's where the kind of phenomenon that you're talking about, I think, come in. So we look at things like advection and diffusion of these solutes. So, for example, let's take oxygen as an example, through the bloodstream and out into the surrounding tissue, and then we capture things like metabolism by the cells. If it was drugs, it would be, how those drugs get transported both intracellularly and extracellularly, how they interact with other chemical components. So absolutely, these are all essentially encoded in the solute transport models. So they're all kind of systems of partial and ordinary differential equations that follow transport phenomena. So very much so, and that's a really challenging and interesting part of the work, I think, because again, it requires the modelling to be really closely linked with dedicated experimental work, either kind of in vitro or in vivo work, to really understand and quantify a lot of these mechanisms. So, for oxygen, for example, we always write down things like Michaelis-Menten to capture the metabolism of oxygen by cells, but obviously there are scenarios where that is an oversimplification, and the parameters in those relationships vary significantly depending on cell type. So pinning down a lot of these features for an individual example is really, I think, important to make the simulation results predictive. So we try and, again, embed all of that in the workflow, if you like.
Thank you, Rebecca. I have a question just building on Huidong's question, which is your modeling and imaging is you're investigating things going from the vasculature towards the outside. I was wondering if this technology that you're developing can be used to also learn from things what's happening inside the blood vessels. For example, the permeability of the blood vessels, or perhaps quality of the blood that is flowing through them.
Yes. Yeah. No. Absolutely. There's obviously opportunity for further development of the frameworks, but at the moment we have the properties of the blood vessel walls captured in there essentially through things like vascular permeability, either to blood or to solutes. So that essentially defines the coupling between the models in the blood vessel networks and we've got Poiseuille flow in tubes to describe the flow in the blood vessels, and we've got kind of Darcy porous medium type equations to capture the flow in the interstitium, and then they're coupled by relationships which, like Starling's law, which essentially capture this vascular permeability. So you get a flux of flow across the vessel walls. So that's how we do that at the moment. You can incorporate lots of different effects like, oncotic pressure contributions and all that kind of stuff in there too. And then the blood flow models are, so we try and keep it as simple as possible because of the computational tractability. But we use Poiseuille's law, and then essentially we use the relationships and the work that's been done by people like Axel Priest and Tim Seckham in microcirculation modelling over the years and hemodynamics, to look at or to incorporate things like how the blood viscosity depends on the hematocrit, so the red blood cell fraction, what happens at, when blood vessels split and bifurcate and all these kind of things. So it's a really active area of work, so there's lots of opportunity, but yes, that's all kind of embedded in the frameworks, if that makes sense.
Great, thank you. Huidong also asks, so the question in general says, how to convert fundamental research into the deployment of technologies in healthcare. What kind of skills do you think more crucial for medical engineering students to enable this?
Yeah, that's a really good question, and it's one that we try to think about an awful lot. So I think for me, what's really important is that if you're thinking about training, which I think is this question's around, but you need to have disciplinary expertise. Obviously, it's really important that we have fantastic mechanical engineers, for example, who have that real depth of knowledge in areas like computational modelling, engineering design, etc. But I think equally important is the ability to work with other people and to work with people with completely different expertise. So, if you're talking to a clinician, they're not going to know a lot about the details of the models we're developing or be interested in the equations necessarily. So there's a lot of skills that I think are really important to embed from a really early stage in our training, that's around communication and understanding problems from the point of view of an end user, and really being prepared to sit down and talk to those kind of people. So it could be patients, it could be anyone, right? People who are going to use the technology. We need to engage them right from the beginning and understand exactly what the objectives are of what we're trying to do. So I think in terms of skills, there's a lot of the kind of soft skills which are really important. So communication skills, and it could be more diverse than that. It could be things like policy work as well. So we're trying to embed things like this in training for PhD students and postdocs here at UCL, so they get the opportunity to do that from a very early stage.
Okay, great. I have another question from Abd El Rahman, and this is related to the last project, the Ventura project. And the question is, do you see a future looking at existing medical devices to help generate new medical devices? So basically repurposing historic devices instead of creating new ones from scratch.
Brand new ones. Yeah, it's a really good question, and I think my answer is probably changing in time actually. So I think in terms of a pandemic response, it was clear that there was a real role for this kind of approach, and I would argue very strongly that that's still there. I think there's a lot that we could do by understanding and adapting devices that maybe have been developed historically. I think the challenges there are around regulation. So, obviously we got regulatory approvals for our devices from the MHRA. The MHRA kind of pivoted their whole processes to develop processes to maintain that safety and efficacy were guaranteed. But that whole process was a lot faster. So our devices, for example, can be used just on COVID patients, and we renew that derogation in line with national need when it's needed. But longer term, we're applying for CA marking, for example, and it becomes really quite complex. So I think there is a real need, but I think in order for that to be successful, we need quite a lot of potential innovation in things like regulation and how we think about that globally as well. So I think the other real challenge is that, of course, we develop medical devices not for use in just one country, right? That you do that to be used in all sorts of different countries across the world, and for that you need to understand the kind of local clinical environment, but also the regulatory landscape globally is really very fragmented, and I think that's a real challenge too.
Can I ask you one last question, Rebecca? Also from this last example, in what do you think were the key elements that made it possible, and what are sort of the take-home messages that you would take from this experience on the Ventura project as we prepare for, say, a potential future pandemic?
Yeah, I mean, I think so. If I think of the things that we massively benefited from and meant that we could move really quickly, it was this kind of culture of collaboration across disciplines, but also with hospitals and with industry. So all of the people that we worked with, we already, for the most part, we already had links with. We were already working with. So and we really benefited from things like long-term investment in common infrastructure across universities and hospitals, for example. So I think there's a real need for funding in those areas. And then, I think forward-looking as well. I mean, I'm going to go back to the regulation aspect. I think that's an area that we need to focus quite a lot of effort into. The MHRA were absolutely fantastic. But, I think, so Brexit is obviously posing quite a lot of challenges for medical device regulation in particular. But if we were to make the most of it, then it's an opportunity to think about how we would kind of redefine that to optimize the regulatory process and understand, this whole deployment. So there's one thing which is developing a device and testing it in, for example, first of human trials, but then that's exceptionally challenging in itself. But then being able to take that forward as commercially viable device that can be deployed at scale is it's an extremely complex pipeline.
Yeah.
So I think that's something to revisit.
Thank you, Rebecca. So that's the end of the questions. We're out of time. I do want to mention that there's a message from Dr. Sylvain Jamet, who is the chair of the Biomedical Engineering Division. Just wanted to thank you for the interesting and inspiring presentation. And again, from me, it was wonderful to have you, and thank you for not only your presentation, but the work you're carrying out. So thank you very much, Rebecca. And thank you, everybody.
Thank you so much.
Yep, thank you. And thank you, everyone, for joining us. I hope you have a nice rest of the day. Bye-bye.